Hello, how are you? Today, inshallah, we will take chapter 11, lesson 2, applying Mendel's principles. Let's start with objectives. The first objective today, which is explain how genetics use the principles of probability to make Punnett square, and objective 2, explain the principle of independent assortment. Objective 3, explain how Mendel's principles apply to all organisms. Let's start with the first principle, which is probability and Punnett square. What do you mean by probability? The probability is the chance of happening. So if we have a tall plant, the tall plants have two alleles. What's the probabilities that the gamete will take the dominant allele, and what's the probability that the gamete will take the recessive allele? So whenever Mendel performed a cross with pea plant, he carefully categorized and counted the offspring. Like whenever he crossed two tall plants, which is the F1, okay, they are hybridized, or as we can say heterozygous, because the parents, or the first parent is T capital and T small. So it is either we can call it hybrid or we can say heterozygous. Okay, at this chance we have to segregate according to the last principle. So we need to segregate, we need to separate the gametes, and the gametes must be T capital and T small. Okay, so after the mating between these two parents, the offspring should be 3 tall and 1 short. Okay, 3 tall and 1 short according to the dominance rule. Okay, in this chance we need to count and find the probability. 3 out of 4 are tall, while 1 out of 4 is short. Mendel realized that the principles of probability could be used to explain the results of his genetic crosses. So the probability is the likelihood that a particular event will occur. So we have a chances here. If you throw a coin, so the chance will be 50% for head or 50% for the tail. The chance or probability of either outcome is equal. Therefore, the probability that a single coin flip will land a head up is 1 chance in 2. This amount is half or 50%. The same probability we will use it inshallah with the segregation and in the independent assortment. In this experiment, Mendel cross produced a mixture of tall and short plants. So let's check. Let's make the hybridization. For this is the parent, the first parent. This is the second parent. So we need to in the beginning to segregate the gametes or the alleles. We have T capital here, T small here. Okay? And the other side, the other parent also, we have to segregate. We have T capital and T small. The mating between these 4 gametes, this is gamete number 1, 2, 3, and 4. There must be mating. That will be forming the first one, which is T capital, T small. The second one, T capital, T small, sorry, this is T capital, T capital. Okay. Here we will have T capital, T small, and here we will have T small, T small. This is the first way that you will find this F2 generation, okay? Again, you have to make sure that you will understand how to find the F2 generation. This is the same thing. So there will be here 2 parents, 2 hybrid parents. The first step is segregation, separation of the gametes, separations of the allele, and then mating the gamete. This is the first and second gamete to form the first offspring, and this is the second gamete with the second, the other gamete to form second offspring. The offsprings that have T capital will be tall. The offspring that have 2 copies of T small will be short. So you have to know why. What is the result from the genotype? What's the phenotype from the genotype? So once we have a T capital, the phenotype will be tall. Once we have 2 copies of T small, the phenotype will be small. For the tall, we have two chances, either T capital, or T capital, T small. In this case, T capital, we call it homozygous. And this case, T capital, T small, we call it heterozygous. Okay? And this T small and T small, we also call it homozygous. Okay? So you have to know the genotype, which is this is the genotype. You have to know the phenotype, which is the phenotype. And you have to know what's the name of the genotype and the phenotype at the same time. Is that clear? Hopefully, inshallah, يكون everything clear. Again, this is how we will do the mating. You have to segregate here and to form that mating, okay? Organisms that have two identical alleles for a particular gene, either T capital, or T small, will be homozygous again, and the one that contain T capital, T small, we call it heterozygous. Okay? The physical appearance or the structure, we call it phenotype, which is physical trait, and the genotype, which is the genes or the gametes or even the allele structure. Okay? The other way that we need to use to find the phenotype is the Punnett square. What we call it, or what we mean by Punnett square, which is using the square or the table like this to find the offspring. On this side, we will add the gametes, and this side also gametes for the other parent. And here we will write the offsprings. This is what we call it, the Punnett square. Okay? We will learn how we will use the Punnett square to find the probability of having one of the offspring. Let's take the first type of the Punnett square, which is one factor cross. When we say one factor, we are talking about one genotype or one phenotype, okay? Like the tallness phenotype, okay? Another one, which is large beaks, which is, we call it B capital for the dominant and B small for the recessive, large beak. Let's take mating between B capital, B small, and another one, which is B capital, B small. Okay, let's do it on the Punnett square, miss. This is the Punnett square. For this parent, the segregation will give me P capital, B small. So we will add the B capital here and B small here. For the other parent, the same, B capital, B small. We will add the B capital here and B small here. Okay, let's do the mating. B capital from this side and B capital from this side to give me B capital here, B capital and B small, B capital, B small, B small, B small. This is the one factor cross. Again, this is the one factor cross. The probability of getting large peaks here is 1, 2, 3 over 1, 2, 3, 4 over 4. Okay, and the probability of getting small peaks is 1 over 4. You have to know how to find the probability, and you have to know how to find the offsprings. Okay, is that clear? Inshallah. Okay, this is the same thing, and this is how you will find the offspring. Okay. The other principle that Mendel's applied, which is the independent assortment. So Mendel's wondered if the segregation of one pair of allele affect another pair. In the last experiments of Mendel, we were doing one factor cross. If there is multiple factor cross, is there any effect from one genotype or one allele onto another allele? Let's check. Mendel performed an experiment that followed two different genes as they passed from one generation to the next. So he studied now two factor cross, two phenotype, two genotype, okay? and he studies these genotypes together, okay, from one generation to the next. Because it involves two different genes, Mendel experiment is known as two-factor or dihybrid. You have to know that, two-factor or dihybrid cross. A single gene cross are called monohybrid cross, okay? But we need to know mainly the two factors or dihybrid cross, okay? This is an example for two-factor cross or dihybrid cross. In this experiment, Mendel studies the round yellow peas. The round yellow, which is the dominant, and wrinkled green is the recessive. So look at the picture here. This is round yellow, the shape of the pea seeds, and this is a green wrinkled seeds, okay? So again, round yellow. So R capital for the round, R small for the wrinkled, Y capital for the yellow, and Y small for the wrinkled. This is how he found the mating. We will do, inshallah, the mating after a while. So in this experiment, there will be a segregation for the alleles. You have to know how to segregate two factors alleles. If we have R capital, Y capital, Y capital, okay? If you want to find the alleles for these or the gamete for this parent, we have to know that each gamete contains two copies. 1, 2, 3, 4. This parent will give me 4 gametes. Each of these gametes have one copy of the first gene and second copy from, or other copy from the second gene. So we will take here R capital in the beginning and Y capital. All of the gametes will be similar here because the alleles are similar. There is no different allele because it's homozygous gene. Homozygous mean two dominant. So all of the gametes will be R capital, Y capital. Okay? And that these gametes will be here. We will put these gametes on this side of the Punnett square. And as you can see here, the Punnett square is large compared with the previous Punnett square, okay? And here is the second parent and the gamete of the second parent, which is R small, Y small. Here, you have to make the mating. All the offsprings here are R capital, R small, Y capital, Y small. You have to know that because the parents are homozygous, okay? So you have to find the gamete in the beginning for the first parent, the gamete for the second parent, and the offspring, which is found in F1 generation. Okay, is that clear? Let's check the results. This is again the generations, the F1 generation, which is the two-factor cross F1 generation, filial generation. Okay? The wrinkles green peas had the genotype of R small, Y small, Y small, which is homozygous recessive, that we can't see in F1 generation. All of the F1 generation is R capital, R small, Y capital, Y small, which is round yellow seeds. What will happen if we want to find the F2 generation? In the last picture or in the last part of the example, we found the F2 generation. Here we want to find the previous one, F1 generation. Here we want to find F2 generation. So all of the offspring were R capital, Y capital, Y small. If you want to find the gametes, each of the allele will take one of the other gene, okay? Allele, of the other gene allele. Let's find the gametes. We will write the gametes here. The first one, R capital. The other one, Y capital. The second one, R small, Y capital. Here, R capital, Y small, and here, R small, Y small. In the same time here, we write the other gametes R capital, Y capital, R small, Y capital, R capital, Y small, R small, Y small. For the offspring, you need to find the mating, R capital, Y capital, Y capital. This is the first offspring. You have to fill in all of the Punnett square to find out the last one, which is R small, Y small, Y small. So your answer will be correct if you find the first one and it is R capital, Y capital, Y capital, and the final offspring, which is R small, Y small, Y small. By this experiment and by finding all of this F2 generation offspring, Mendel's found that 315 of the F2 seeds were round and yellow, while 32 seeds are wrinkled and green. Okay? This will indicate that the relationship between the round and yellow and the same time green and wrinkled will be independent assortment relationship. That will follow the independent assortment relationship. So again, the alleles for seed shape segregated independently for those for seeds color. Okay? Genes that segregate independently, such as the genes for seed shape and seed color in pea plants, do not influence each other's inheritance. So after he finished this experiment, he concluded that there is no relation between these two alleles. Okay? Also, Mendel has discovered the principle of, again, what we call it, independent assortment. The principle of independent assortment states that genes for different traits can segregate independently during gamete formation. So, Miss, you have to memorize this principle, okay? You have to memorize it, okay? And that's it. Thank you and have a nice day. Try to do more examples, try to do more questions on the independent assortment. I will attach you a homework about the independent assortment and try to solve it and give it to me at the class. Have a nice day and thank you.